Hello, everybody. Good morning. Thanks for being here today. I'm very glad to introduce Professor Patrick Keeling from UBC to today's Friday talk. Patrick is a protozoologist. He's an expert on many things, but he has been working on symbiosis for many, many years. And I was lucky to spend some years with him in his lab as a postdoc. And I thank him a lot to be here today, and well, enjoy the talk.
Thanks. All right, can everybody hear me with this thing? Is it good? All right, good. Well, thank you for the introduction, and also thanks for, I sort of invited myself, so I appreciate the chance to be here. And I'm going to try to give a little talk here that touches on some things about marine biology and evolution, recognizing that there's two different institutions here, so hopefully that works. Let me just make sure this works. Excellent. Okay, so what I want to talk about today is organelles and endosymbiosis and evolution, but I want to talk about, instead of the origin of endosymbiotically derived organelles, I want to talk about what happens when they disappear. Because to understand the evolution of one of these things, you need to know how it originated, and we spend a lot of time thinking about that. But I'd argue that you also need to understand what happens when they vanish, because to look at the distribution of these things on the tree of life and understand the presences and absences, you have to understand gain and loss. And we don't actually know much about loss of organelles, partly because it's very rare, but it's also a bit of a blind spot. Okay. So this is a tree of eukaryotes, and mitochondria are present almost, but not quite everything, and plastids are scattered all over the place. And I'm going to talk about plastids because there's way more gain loss, there's really interesting evolution. But as I said, they are kind of scattered all over the place, so just to keep things a little bit simple, I'm going to talk about a single lineage, which is the mesozoans, which are a subset of the alveolates, which are, one of those protist groups that most times when you give a talk, nobody knows what you're talking about. But like everything with protist diversity, like this is a very, this is a big tree of a huge amount of mostly protist diversity. But when you look at even a lineage like this, like a subset of it, it's almost like a fractal, because when you start digging into that, you find the diversity just gets bigger and bigger and bigger, and then you dig into subgroups and it gets bigger and bigger and bigger. So this is a blow up of that group. And as you can see, there's two main subgroups, the dinoflagellates and the apicomplexans. And each of those has many, many subgroups with lots and lots of diversity of morphology, of trophic types, ecological impacts, and whatnot. But this is a great playground for studying molecular evolution, like the gain and loss of really, really interesting evolutionary characteristics. And one of the reasons I enjoy it is because their ancestor was photosynthetic, so that it had a plastid, but it was probably mixotrophic because it was probably eating by this thing called mesocytosis. And one of the things that duality allowed was lots of losses of photosynthesis. Photosynthesis has been lost many, many times in this group. Some of them are ancient events, which impacted a huge lineage, and lots of them are less ancient events that impacted a subset of the lineages, but they're all the same event. They're all the same loss of photosynthesis. So I might say this one's bigger, but that's actually a dumb thing to say, but I say it all the time. It's actually the same event. It's just it happened so long ago. So I want to talk only about a couple of these. The first couple really quickly, just to give you a taste of the kind of things you can do with these. And I'm going to spend more time on this one here later. But let me start with the dinoflagellates and just one little event in one little group, just to give you an idea how cool this can get. So this is Liz, PhD, now postdoc in my lab. And when COVID struck, she didn't give up. She stole this microscope, and a plankton net, and she went down to the beach every morning and did plankton toes and started isolating single cells. These are just some of the cells she's isolated. She's now isolated over 800 cells. And for every one of them, she has pictures and movies, and she sequenced its transcriptome, so the bulk of all expressed genes. And she can get 30,000 genes out of each cell. So this is a fantastic amount of data that has bankrupted my lab, but there's a lot of neat stuff here. So for instance, she noticed these cells have these black dots. and others in this thing do too. And she recognized what those black dots were, and that these cells were particularly interesting, because these are Warnoweoids. So here's a Warnoweoid, and you can see the black dot there. You're probably immediately drawn to this thing, which is called the piston. And unfortunately, I'm not going to tell you anything about the piston because nobody knows how it works, what it's made of, or what it does, what it's for. It's completely mysterious, but it's fantastic. This is the kind of thing that I love about protostology. The black dot is actually part of an eyeball that's incredibly complicated with a lens and a retina and a cornea that are made of subcellular organelles. This is an old characterization from a co-supervised student. You can see here this is the equivalent of the retina, and what he showed is it's actually a non-photosynthetic plastid. And it was thought to be driven by some sort of proteorhodopsin light harvesting proton pump, because our eyes use opsins. But proteorhodopsin isn't usually in the plastid, and so we really didn't know anything about how this eyeball worked. We didn't really even know it detected light, because there's no functional data from these things, because none of them are cultivatable and there's no genomic data. There was a transcriptome with a Busco score of 0. If you do transcriptomes, that's really, really bad. So we have no functional data from this thing because we can't grow them. So Liz picked, I don't even remember how many of them, maybe 20 of these cells, because they're not very common, which was every single genus known, plus she found one new genus that we didn't know about, and sequenced multiple cells from each one to establish a bit of functional information about how this thing works. And what she found is one of these things in retrospect shouldn't have been that surprising. Oh yes, these are the cells, these are the phylogenomic tree. So you see she's got a nice distribution of them with multiple cells from each one. And when she dug into what the plastid's doing in these things, what she found is the expectation would be that non-photosynthetic ones don't have any photosystems and photosynthetic ones do. And what she found instead was that they all had some photosystems, but not all of them. Specifically, they lost photosystem 2. And this is a physiological state that lots of algae get themselves into when they're light stressed. It's called cyclic electron transport. And it allows you to do a little bit of ATP synthesis without the danger of splitting water, which is the really horrible step in photosynthesis that makes lots of reactive oxygen species and can kill you. So these organisms have adopted this permanently, and they're not actually doing photosynthesis with it. They're just using it as a light-driven proton pump for a sensory mechanism. And if you think about it, we should have thought of that right away, because the chloroplast already has a light-driven proton pump. It just uses it for photosynthesis. So if you get rid of photosystem 2, you make it a safe way to use your plastid like we use our retina. And that's all they've done. They've lost this photosynthetic complex, they've lost photosynthesis, but they've kept the rest of the mechanism basically as a sensory organelle. So they can probably chase down other algae and hunt them and eat them, which is what they do. They shoot out these complex ejectosomes and paralyze their prey and engulf them. So they're not algae anymore, but they've kept their plastid for this other purpose. And we're able to say this because these are big complexes, and you couldn't do this with one single cell, but you can do it with 5 genera with 15 cells each, because you can see the presence and absence patterns quite clearly from that kind of data. Okay, I'm going to move on and talk about these losses here. And these are these are interesting because of the ecological impact of these cells. So these are called malves, or marine alveolates. And the reason they are called malves, and they don't really have a proper name, is because we don't know much about them. But this is interesting. The very first time anybody ever did like a survey of marine diversity of protists in the ocean, they found these things right away because they're so abundant. Right, like 20 years ago, the first of these surveys came out in 2 papers in Nature, and immediately sort of said, oh my gosh, most of the diversity is these unknown things, which are somehow related to dinoflagellates. And I picked this, actually I didn't pick this because I'm here, I just like this paper. So this is an example from you guys, a fairly recent one, and it just shows you what I mean, like MALV 1, MALV 5, MALV 5, MALV 2. It's a huge amount of eukaryotic diversity from marine surveys are consistently these things called MALVs. And yet we've hardly ever even seen most of them. We've got a couple in culture from way back, but we don't even know what these things are, and yet it sometimes is half the diversity of our sequences from the ocean, and we don't know what they're doing. They're assumed to all be parasites because the few that have been examined are parasites, so that makes sense. But we don't know much about them. Now, the malves are broken up into many different subgroups, but they're mostly in two major sets. Malv 2 and its relatives, and Malv 1 and its relatives. And thankfully, just by chance, the way they've been numbered, the odd ones tend to go together and the even ones tend to go together. So I'll talk about even malves and odd malves. And these are two big lineages who have been proposed to be related, and they've been named the Syndineales in the dinoflagellates, after one of the few organisms that we actually know about. So Syndineales, malv evens and odds, very interesting, particularly because two genomes have now been done from a malv 2 and a malv 4, and they have completely lost their plastid. And this is very, very rare. And it's very, very rare because you have to know that something doesn't have a plastid and isn't just non-photosynthetic, but you also have to know that its ancestor did, which is even harder to know sometimes if you don't know the phylogeny. Now, this is very exciting, a glimpse in plastid loss. However, it's only half the story because these are both malv evens. So we don't know about the malv odds. What are they like? And many of the little subgroups, too. And that's because there's no genomic data from any of them. There's no cultures, not an easy way to get it. So we did actually a couple different things to get at this. First of all, we found free-living relatives of malves that aren't parasites. So that's important to understand the origin of parasitism. And we got those in culture, and that was largely Dennis, who many of you might know is an ace at culturing things. And we also used single-cell transcriptomics to get parasitic malv 1 transcriptomes. So we can add the malv odds to the equation. And these are just some pictures of the things. This is one of our single cells, and you can see this fried egg inside this dinoflagellate here is a malv 1, and these are free-living things that are related to malves. So what's interesting, though, is when you put it into the picture, it turns, or into the phylogeny with phylogenomics, malves aren't a lineage. So Syndinians are not a lineage. They're two different lineages. So the malv evens are one branch, and the malv odds are a different branch. They did not originate in from a common ancestor, and that means that their parasitism is also arose twice independently, even though they have what appears to be similar kinds of parasitism. It evolved twice, and we're very certain about that because in both cases they're related to a free-living relative. So the malv odds are related to Oxyrus marina, which is a super common, like tide pool dinoflagellate relative, and the malv evens are related to these new things that we discovered called alephtherids. And so we know that their parasitism evolved twice, which is already very cool. So then the question becomes, well, what about the plastids? Well, it turns out malv 1s have not lost their plastids. They still have plastids that are not photosynthetic. And so their common ancestor was probably non-photosynthetic, and then they evolved parasitism. But what about up here? So here we have this case of complete plastid loss, and now we have a plastid-bearing, free-living, non-photosynthetic thing. But it raises the question, what happened first? Did they become parasites first, or did they lose their plastids first? And you can't really tell from this data, because those two things go to the same place in the tree. So this means back to the field to find more stuff. And Liz, once again, got invited on this expedition, this NSF boat that was actually created for research, so it's a really nice boat, that goes out of Alaska on a long-term cruise, and we're hoping that keeps happening because she's been invited on this now every year, and she goes out and does her single-cell thing from deep plankton tows, way deep plankton tows, out in the Gulf of Alaska every summer. So she's got a nice little lab set up in the boat, and you can monitor when the material's coming up, and she can get fresh material right out of the deep sea and start picking cells and freezing them for transcriptomes. And one of the things she found is really interesting in a lot of ways. First of all, it's a collidarian, which is something that we don't study very much. They're really interesting members of the radial era. That's not really a thing anymore, but whatever. And these were infected by some strange-looking parasites, and so a bit of a double whammy here, because collidarians are cool by themselves, but collidarians that are parasitized are really cool. So she isolated lots of these cells, did lots of genomics on it, And she found a new Malv even lineage, which we're quite happy. We're on 6, so it actually worked out perfectly, so we can keep the even and odd. So Malv 6 is this new kind of parasite, and it branches actually within the Malv even clade, although I wouldn't rule out the possibility that it's a sister to the other Malv evens. And what's really exciting about it is that it actually hasn't lost its plastid. So Malv 6 does have a plastid, and it appears to use it for isoprenoid biosynthesis and iron-sulfur cluster biosynthesis. And so it's kind of nice because it can give us a window into, at least possibly, we can now say that probably parasitism evolved, and then after that, in a subgroup, or maybe twice, the plastids were lost. Although, you must have noticed by now that parallelism is becoming a big theme, and it really is a theme with the origin of parasites. Many things that look like they're complicated that should happen once have happened many times. So you have to be a little cautious about these kind of things. And I'll get into that in detail here. Because now I want to talk about this group, the origin of apicomplexans. Good. So, oops, how do you go back? Apicomplexans are well studied, much better than dinoflagellates because they're all parasites, the entire group, mostly intracellular, mostly parasitic, but they're obligate symbionts of animals. And the most famous one, of course, is malaria, the malaria parasite Plasmodium. And many years ago now, malaria got really interesting when they found that it had a plastid. And so the Plasmodium plastid, was, the parasitologists that discovered it just took a beating from the field because everybody said that's so stupid, they can't have a plastid, blah, 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 blah. Of course they do. It's very interesting, and it traces back to this common ancestor here. But it's meant that this plastid is actually really well studied. It's by far the best studied non-photosynthetic plastid now because malaria is an important disease. So I got interested in this ages ago when I worked in Jeff McFadden's lab, and that's actually what got me into apicomplexans. And I was more interested not so much in the function, but in the evolution. Like, why does this thing have a plastid still, and where did it come from? How did an alga turn into an obligate intercellular parasite of human red blood cells? It's an interesting question for an evolutionary biologist. And there's a big breakthrough shortly after I started my own lab. This is ancient history, just to sort of set the stage for this. With this discovery by some researchers in Sydney of these two organisms, Chromera and Vitrella, that were coral symbionts and were thought to be normal coral symbionts of the Symbiodinium type, but actually when they looked at sequences from them, these little brown balls were different little brown balls that actually branched at the base of the apicomplexans and not with the dinoflagellates. And so, very exciting. Now we have these relatives of apicomplexans that are still photosynthetic. So a nice little missing piece of the puzzle there to study. My lab got involved because we sequenced the plastid genome from these things to try to work out if that helped us understand the apicomplexan plastid, and it certainly did. It showed us that they're red algae and solved a whole bunch of things that people have been arguing about for years. That's nice. But like other good projects, it actually raised a whole bunch of interesting new questions, and that's what I want to talk about. So this is not our work. This is like a pretty standard kind of microbial ecology study with lots of ribosomal RNAs sequenced from some environment, in this case from coral reefs. And here's a novel phylum of bacteria that were frequently discovered in this sort of time period. It doesn't branch with anything in particular, although it has this really interesting association with cyanobacteria, but it was just called a novel bacteria. But of course, this was before we did the plastid genomes from Chromera and Vitrella, and what we discovered is that these aren't bacteria at all. These are Vitrella plastids. But because that genome wasn't available to these researchers, they had no reference, so they didn't know what it was. But it led us to start to ask, well, how many times has this happened? How many plastids are there out there that have been called novel bacteria? And how much of this apicomplexan-related plastid stuff is there? So we did a couple studies on this, and basically we looked at other people's work from all over the world, and we pulled out all the plastids that have been misidentified as bacteria. And there's a lot of them, hundreds of thousands of them. But what we found is basically they were all known groups of algae. So they actually fell within the group. So they were within the dinoflagellates or within the diatoms or within the green algae. They weren't like some mystery thing that we didn't know what it was. And the few exceptions to that were all related to apicomplexans. So they were the sister group to the apicomplexans. They weren't in the apicomplexans, they were just the sister to it. And they were also basically all associated with coral reefs. And this is important because coral reefs are well studied, but they were not overrepresented in this data. It was mostly open ocean, planktonic. So there's, about close to 100,000 of these things with tens of thousands of species. So it's very diverse too, and they were split into several different subgroups. The most common subgroup was called Arrow5, and we decided to go after that because it was quite common and it was very closely associated with corals, coral animals, not just the reef. But we don't know anything about it. So, at that point, you're like, does it live in coral? Is it a symbiont, or is it on coral? Is it photosynthetic? Don't know. It's just a ribosomal RNA from the plastid. So we know it has a plastid, but we don't know if it's photosynthetic. Alternatively, are they parasites? Do they cause disease? Loads and loads of questions that required field work. I was actually at a stage in my career where I was really sick of sitting in my office and answering emails. And so this was very appealing. And so we went off and started this field program in Curacao. I would like to tell you that we stayed at this luxury resort, but we did not. We stayed in the bunkhouse at this research station. But it's a fantastic place, mostly because the director is great, Mark Vermey. He knows everything, and he's so helpful. And he got us in, he and Forrest Rower got us into field work, where we actually went out for a few years. John Steinbeck has that book about the Sea of Cortez, where he says that you really have to do everything twice. The first time you make all the mistakes, and then the second time you get the data. I would disagree. I think you have to do it about 10 times. So we kind of went out and sort of taught ourselves to do field work, but this is a great place to do it because here's the dock, here's the coral reef. It's an interesting place to go, and it was really very highly impactful for my work. So over the course of several years, we chased down this Arrow5, which we now call corallicolids, and we can now see them. Here's some TEM, here's some fluorescence showing that they live inside the mesenterial filaments of many, many, many kinds of coral and related species. This is Walden Kwong, who really pushed this work, and he sequenced the complete plastid genome of a corallicolid. Sorry, is that okay? Okay. Right. And so the plastid genome is incredibly informative because the first thing it showed us is that corallicolids are definitely non-photosynthetic. They're not a photosynthetic symbiont like Symbiodinium because they have no photosystems. And the photosystems just can't all be moved to the nucleus, probably because of targeting. So we know already that they're not photosynthetic. But unlike every other photosynthetic plastid, including those of other apicomplexans, they're very odd because they kept all the genes having to do with chlorophyll biosynthesis that the plastid genome would have had ancestrally, which in this case is 4. So that's really weird. In fact, at this point, it's unique. Nobody had ever seen a non-photosynthetic thing that made chlorophyll. And if you think about it, why would you? What a dumb thing to do. Because that is incredibly dangerous. Chlorophyll by itself without it's like having a bomb without a way to defuse the bomb. And so this is a problematic thing to study, but this is what we want to get into. And it's also complicated because we didn't actually know where they went in the tree. So if you look at the nuclear ribosomal operon, it puts them here with this group called coccidians in the apicomplexans, so what we'll call a sort of late version, like a recent origin. Whereas if you look at the entire plastid genome, in fact, puts them really deep in the apicomplexans, in fact, sister to the apicomplexans, so that's like a very early origin. And the chlorophyll thing kind of makes more sense down here because this is where photosynthesis was lost, somewhere around here. So it would imply that chlorophyll was kept and photosynthesis lost, but only, there's only one node difference. And so it was kept in this lineage for some weird reason and then lost once here. So I have to admit, I sort of thought this was going to turn out to be right, and Walden thought this was going to turn out to be right, and we argued about it a lot. But we didn't have the data to tell because neither of these is conclusive. You need to get genomic data to tell that. Furthermore, we only have 4 of the chlorophyll biosynthesis genes, and it doesn't include the last one, which is the one where it turns, protochlorophyllide into chlorophyll. So we can't really say they have chlorophyll at this point. We can just say there's these 4 genes, they're under selection, and they're expressed. So there's a lot of stuff that you'd need to get the genome for, but that turned out to be really hard to do. We went back to the field on 5 different seasons, trying to figure out how do you get these obligate intercellular parasites purified enough to get genomic data from them. So we identified 2 really great hosts, a stony coral and a zoanthid. We worked out ways to do Percol gradients in the field where you could sort of separate host tissue from highly enriched parasites that still have host tissue. By the way, these don't have symbiodinium, and these are very, very low in symbiodinium. They're mostly heterotrophic corals, so that helps too because you don't have a bunch of dinoflagellates. And then, we could see the parasites on the light microscope, and we got some more pictures and stuff like that. But we also got these highly enriched fractions where we could sequence transcriptomes. And this is Victoria here working on it, and she got the transcriptome data from both these hosts. And it's very nice transcriptome data, huge representation of all the genes you'd expect to find in an apicomplexan. And we got a very, very conclusive phylogenomic answer that actually corallicolids arose late. So they're fairly closely related to coccidians, perhaps specifically related to this protococcidian group. Of course, there's now another group in here, which Javi's lab discovered, which are the ichthyocolids, which branch here too. And that actually doesn't really... We sort of threw them in at the last minute here, and we showed that it doesn't change the overall picture, but they are indeed related. So, what about chlorophyll? Oh, actually, I threw those slides out. I'll just tell you. The data also show that the entire chlorophyll pathway is there, including the final enzyme, the definitive enzyme in the pathway. So these things are definitely making chlorophyll. The enzymes are all expressed. They're all under purifying selection. But, I didn't throw them out, but now I don't have to say it. Okay, so then I just want to back up now and think, so what do we now know about this whole group in plastids and photosynthesis and chlorophyll? Because when you lose photosynthesis, you almost, in every other group around the tree of life where photosynthesis has been lost, you lose chlorophyll at the same time, right? Except this group. So what do we know now in this group? So if you see this tree, there's 3 occasions where you see the black line. Those are things that have completely lost their plastid. So the gray line represents things that have plastids. And so plastids have been lost 3 times. That's already fantastic because I like looking at plastid loss. But if you look at photosynthesis, it's now we can say, been lost at least 5 times because there's a couple of photosynthetic relatives here. We know the ancestor is photosynthetic, so there's all these cases like the squirmids and all these apicomplexans where photosynthesis has been lost. So it's a total of 5 times. So that's a lot of parallel losses. But if you look at the position where corallicolids are in the tree and the fact that they have fully the entire chlorophyll biosynthetic pathway and it's all expressed and under selection, it means that chlorophyll was lost, I think when you throw in ichthyocolids, 11 times. And so here's the event here where photosynthesis was lost, but chlorophyll wasn't. And as I said before, if corallicolids had branched at the base of the apicomplexans, this would make a lot of sense, but they don't. They branch much later, which means that every single apicomplexan lineage other than them had to independently lose chlorophyll. So we still don't know what chlorophyll's doing, but this actually tells us something really important that narrows down the kinds of things it could be doing, I would argue. Specifically, I don't think it can have anything to do with ecology. Because when this was first discovered, these things are symbionts of corals. And everybody's kind of thinking like symbiodinium, photosymbiosis. They're thinking, this corals, they live in the sunlit, shallow seas. So maybe it has something to do with that. They live in this high light environment. Maybe they needed chlorophyll to protect themselves from the light or whatever. But I would argue that's impossible because all of those ancestors down the entire backbone of the apicomplexans almost certainly didn't live in corals. They probably didn't live in, shallow sunlit seas either. So I would argue that there must have been so many different ecological contexts down that backbone of the tree that whatever it is that the chlorophyll is doing more likely has to do with cell biology. So it's probably something really fundamental. And I'll give you an example. And these are, I should say, boring things. They're not like exciting, like protecting you from sunlight and corals. So for example, the chlorophyll is part of the heme biosynthetic pathway. The heme pathway leads up to the chlorophyll pathway. So if you had a feedback inhibition from chlorophyll to turn off heme biosynthesis when you had too much chlorophyll, for example, and then if you lost photosynthesis, you might need to keep synthesizing a bit of chlorophyll to control your heme synthesis until you came up with a different way to inhibit heme synthesis. And that would be the kind of thing that would be easy to come up with many times independently. So currently, that's my favorite hypothesis, is that it's just a relic of an ancestral feedback inhibition to control heme biosynthesis. Of course, that's very hard to test because we have these fairly rare parasites that are obligate intercellular parasites of corals and other anthozoans. Okay, how am I for time? Oh, good, lots of time. Because I want to end with a little COVID story. Of course, COVID made a big mess of this because we're right in the middle of a bunch of field trips to Japan and Curacao, and they all got canceled. And I had this project that I wanted to get done, and I was trying to get Walden to do it, and Walden was, like a senior postdoc, so of course he thought I was an idiot and said no. But I think it was a good idea. And it has to do with how we do field sampling and biases. So I don't do this, so I'm not going to, try to put words in anyone's mouth. But we do a lot of sampling where we go out in the ocean and we filter things and we look at different fractions and we sort of make stories up about, not make up stories, we infer what's going on from the different fractions. And we've developed these methods to sample these fractions based on what tends to dominate them. And we do a lot of sampling of the plankton. And, when the stuff that I think is really cool is when you get down into the sediment, but that's really hard to do when you're out at sea, so it doesn't get done as much. So we've got tons and tons of data from the plankton. We've got some data from the sediment. But one of the things that happens is we tend to separate the animals from the microbes, and then we study them using different methods. And so you don't actually really sample parasites, different kinds of parasites at the same level. So let me give you an example. If you went to Africa and you wanted to know if malaria was there, and you filtered a lot of air and maybe some soil, and then you filtered out all the animals and sequenced all the protists, you would not find malaria, right? Because it was in the mosquitoes that you'd filtered out. And then if you studied the mosquitoes using a different method, you wouldn't know malaria was there either. So you would conclude that malaria didn't exist. And so what if that happened here with corallicolids? Some parasites have a very open infection system. So this is a malve, which we find all the time in the ocean. This is Amoebophria, and it has this vermiform. It's coming out of a dinoflagellate that it's consumed and killed, and it makes this huge multicellular worm that then spirals away and swims out into the ocean, breaks up into thousands of cells and gets eaten by more dinoflagellates and continues the infection. So that's a very open infection cycle, and that's why malves are all over this tree, because they're barfing out huge amounts of these infectious particles that we're picking up in the protist-size fractions and characterizing. But what if corallicolids are all over the place, but they don't do that because they have a closed infection cycle like malaria? Because they're pretty closely related to malaria, to be honest, so why not? And so I was arguing that our own data might be wrong, that this is actually the bias that they're associated with coral reefs is just our bias because it's one of the only invertebrates that we actually look at a lot is coral, because we love coral. So I was pitching a project to go look at anthozoans that aren't on coral reefs, for example, and of course everybody's like, ooh, I want to go to the coral reef, right? Nobody wanted to do this. So during COVID, though, we didn't have much choice. And we developed this little low-tech thing without a lot of world travel. And an undergrad here, Morelia, she took this on and she loved it. She sort of made this project her own. So it was supposed to be like this little thing, like, let's just go out and see if there's something there. And she ended up collecting like 350 anthozoans from all over the coast of British Columbia. And we have lots of anthozoans. It's not, we have coral, like this is a stony coral. It's just not a reef-building coral, but these are all from British Columbia. So there's a coral. We have lots of anemones, way more than you find on a coral reef. We have zoanthids, which is one of our model systems from the coral reef. We have corallomorphs, which are one of my favorite animals and really overlooked animals that are very cool. They're the sister to corals. And we have octocorals. This is a sea pen. And so we have all the different subgroups, except we can't get to black corals because they tend to be really deep in the cold water. And as I said, she did this massive survey of them. And what she found is that corallicolids are all over the place in British Columbia. They're in virtually all the anthozoans that we could find. So on here you see the red things are tropical coral reef corallicolids, and the blue ones are cold water anemones. And so that's cool. We now know that corallicolids aren't on coral reefs. They're just in they're in anthozoans. But the distribution, you'll notice, isn't quite random. Like this half of the tree is mostly red, this half of the tree is mostly blue. And so it raised this other question: is this a cold clade? Weird. And is there a hot clade, or is it an anemone clade and a coral clade? Hard to tell. So we went back into the field. Oops, threw out some slides. To get some anemones from coral reefs, but also to just get some full-length sequences so we could do proper trees instead of these little short things. And what Morelli was able to show is that if you look at all the different characteristics that differentiate where these things come from, which are like which host it's in, the water temperature, how deep it is, and whatever, none of it correlated with the phylogeny at all. The depth might, there's just so little data from deep water things, but none of the other characteristics did for sure. But what did was which ocean they were from. There's basically a Pacific clade and an Atlantic clade. And so this is really interesting because it suggests that the corallicolids can move between hosts really quickly in pretty distantly related anthozoans. So you can get the same corallicolid actually in a stony coral as you do in a zoanthid. So they're bumping around between hosts, and they're bumping around between hot water and cold water actually fairly fast, but they're not moving around the world very fast, which is kind of curious, and we're going to follow this up. And this is some of the stuff we're doing now. So we're trying to go to different, like non-coral reef environments, and that's fun. I'm enjoying this a lot, but we have a little mobile lab that we can take with us, and so you basically have suitcases full of microscopes and scuba gear, and we're going out around the world and trying to get corallicolids from, the Pacific, but in the north, and the Atlantic, but in the north, like just trying to get them from all over the place and see if we can work out any more about the biogeography. The other thing we've started doing, there we go, is, this is supposed to be a movie, is we're going to start looking sort of across eukaryotic diversity a little bit harder at this problem of the sampling bias. So this is a little bilge pump, and it's hooked up to a plankton net and a light and a power source. And you can set these out at night, and they attract lots and lots of animals, and then it can filter like maybe 1,000 liters of water gently through a plankton net during the course of 45 minutes, say. And so then when you come out, you get something that's like this, where you have accumulated a lot of diversity of different kinds of animals. And instead of treating them like animals, we're going to treat them like microbes. So we're going to do like a meta-barcoding sequence with contrametazone primer set to look at eukaryotes and a standard bacterial set to look at bacteria. And we'll get really kind of an amazing database of the animal-associated bacteria, archaea, and protists from this, which is something, we do have some of this data, but we don't have enough of it just because of the biases and the way we mostly do our sampling. And so we've already done a few of these, and we're finding loads and loads of apicomplexans that you would never find in a standard sample of protist diversity. That's the future. We don't have that data much yet. Okay, so lots of people to thank. Victoria really spearheaded the genomics on the corallicolids, and Morelia spearheaded the cold water stuff and now the biogeography stuff. And then these guys, this is the team that did the alephrids and the malve things, and of course Liz also did the Warnoweid study, and then there's a few other people that contributed to all those studies, mostly microscopy. And then I want to thank funding, primarily the Moore Foundation, who pretty much single-handedly fund my lab, so very grateful for that. And 38 minutes, nice. All right. Thanks. I was told to leave lots of time because you guys are really tough on speakers with questions. So.
Okay, it's time for questions.
Ooh, ouch. This is the worst outcome, by the way.
Okay. Wait a second.
You mentioned that it's dangerous to have chlorophyll. What is this word?
Chlorophyll.
Chlorophyll in the cells, but still this lineage has it. Can you explain a bit more why is it dangerous, and what do they do to avoid this danger in the situation that they don't actually use it?
Well, we don't know, so this would just be speculation. And it's actually a little bit difficult. And I'd need to talk to some more chemists about what would chlorophyll do if it wasn't complex with a photosystem. Because, of course, in nature, it's almost always complex with photosystems. And so when it's hit by a photon, it releases high energy. That's the problem that you would worry about. If chlorophyll does that on its own, which I should know but don't know, then you would have a situation where if your cell was full of chlorophyll and it was being bombarded with light, you'd be constantly making probably reactive oxygen species, which are bad for the cell. So my suggestion is they probably do have this problem, but they probably don't make very much chlorophyll, especially if it's just a molecule that's being used in a feedback inhibition or something like what I said. You would have tiny, tiny, tiny quantities of it, and it probably wouldn't be that dangerous, would be my guess. They're not colored, they're colorless. But again, I don't really know what it would be like having, even if they filled the cell with chlorophyll, I don't know what that would be like, because nothing else really does this. So, yeah, it's hard to say. I suspect they're making infinitesimally small quantities of it for some reason, like something adjacent to what I suggested.
Yeah, thanks. Can I ask a second question quickly? So in your last slide where you showed the concentrated animal fraction, did you find any ichthyosporidian sequences?
Ichthyosporidians?
No, ichthyosporia. Basically the protists which are closely related to metazoa.
I'd have to. We literally just got the first of these back it was just a test to make sure it worked. And Marilia is doing this, and she literally just looked at the apicomplexans so far. But we got a very widespread distribution of different kinds of eukaryotes. So I'm guessing we probably would, yes. Groups that tend to be animal associated and parasitic will probably be enriched in this. We just don't have. We haven't had time to check yet.
Thanks.
My question is also related to these hypotheses of the feedback loop between chlorophyll and heme. I was wondering if you looked at the gene expression profile of these pathways in the transcriptomes to see if they follow any specific trend. And also related to that, if you consider the possibility that it's not regulating heme, but, for example, vitamin B12, which also has a very similar structure.
Yeah. Okay, so first of all, I should say that I don't think single-cell transcriptomes are very good for that kind of thing because they're not really quantitative. It's weird. So genes that are very highly expressed will probably appear more in your single-cell transcriptome, but there's an amplification step, and so I'm always really reluctant to talk about whether something's highly expressed or low expressed. We tend to say things like it's very abundant or it's very rare. So there's that. I don't want to overinterpret the data. You'd really want to get some quantitative, like use qPCR kind of thing on an infected tissue would be the way to approach that problem, and it would be a great thing to do. So we've thought about this, like if you take an infected model system in the lab, you could treat it a bunch of different ways and see whether you can find reproducible changes in gene expression. But we haven't done anything like that yet. We could in the field in Curacao. I think especially the golden anemone or the golden zoanthid, the parazoanthus one would be a pretty good model for this. And it's the kind of things we're thinking about doing. There was a second thing I wanted to say though. Oh, yeah, they don't have B12 synthesis, but it's a good idea. But they've stripped out, like they're parasites, so they've lost a lot of biosynthetic pathways.
The host doesn't have it either, right?
Well, that I don't know. I'd have to go check. We did a transcriptome from the host because neither of these hosts have genomes. And we didn't look for that, but we should. It's a good idea.
Thank you. Very cool stuff.
Thanks.
Another question there?
Hello, thanks for the talk. So I have a question regarding the contrast, the disagreement between the phylogeny of that of the small subunit of the chloroplast and the whole organism. Do you have an hypothesis of why it finds such a contrast, and if you try to check if there was also a contrast in the rest of the genome of the plastid?
Yes. Okay. Good question. So why did the plastid genome and the ribosomal operon and the subsequent phylogenomics disagree with each other? I have to admit, I'm guilty of glossing over that because I've been making phylogenies now for so long that it doesn't surprise me when things give you the wrong answer because you just kind of see it all. Anyway, in this particular case, plastid genomes in some lineages are notorious for being wrong when you do the phylogeny from the whole genome. In some lineages, they seem to work fine. In some, they're very misleading. And then people have done really nice studies where they sort of dissect the genome into functional components. So like everything to do with photosystems, they try by themselves, and housekeeping, they try by themselves, or fast-evolving genes and slow-evolving genes, which oftentimes are the same partitions. And what they find is that there's real systemic biases in some lineages, plastid genomes, that lead to phylogenies that are just actively misleading. And so I think that's probably what's going on here, because if you think about it, the apicomplexan plastid genome is quite odd. This is a non-photosynthetic group, and it's been non-photosynthetic for like 1 billion years. And they only keep the genome for one gene. That if it moves to the nucleus, you can lose the genome. So I didn't plot that, but several apicomplexan lineages have lost the plastid genome. Very strange genome, high rates, weird GC biases. So we've sequenced one recently. It's 9% GC. So it's crazy. They're very odd genomes. The ones from Plasmodium and Toxoplasma, the model systems, are quite a bit more normal than that. But still a bit weird, suspicious. So I think they're just probably wrong for the usual reasons with systemic biases in phylogenetic reconstruction. But honestly, we didn't get into it in too much detail. Once we had the phylogenomic data set, it's just so much better. We just analyzed that mostly.
Okay, thanks. And out of curiosity, is it possible to try to do a phylogeny with this single gene that seems to be more conserved?
Oh, yeah, we have done single gene trees on all of the genes. And by the way, I should add that the chlorophyll biosynthesis genes haven't just been horizontally transferred back into this lineage, which is something that sometimes people wonder about too. Because when you do the phylogenies with them, they branch very strongly with the homologs from Vitrella. So it looks like it is the ancestral gene that's still in the genome where it used to be. So each of the individual genes, it's kind of like you'd expect. They're a bit they're a little tricky, but there's nothing about them that screams out that there's anything going on other than some lack of resolution. Yeah.
Thanks.
More questions? One question.
Thanks for the talk, Patrick. Very interesting. I see that you are moving now to try to identify the proteins inside metazoans as a new environment that has not been very much explored in the sea.
I don't mean to. By the way, I don't mean to say it hasn't been explored. I just think it's something we could do more on. Yes.
Do you have an opinion about how much diverse would be the protein composition in a single animal? I mean, it's supposed to be one species or 10 or 100 or many different lineages. Do you have any idea on that?
Do you mean if you take like multiple individuals of a species, how similar will their microbiomes be?
No, meaning in a single animal. If there's going to be a lot of different species, or it's going to be just one. What you have seen with the coral.
We have a bit of information on this already because we did a project a few years ago where we sequenced the microbiomes from 1,000 meofauna from 22 phyla, because mostly microbiome studies have looked at like one model organism, and they're almost always insects and vertebrates. And so we just kind of looked at all these meiofauna. We did deep sequencing of all the bacteria, and we started doing protists too, but we actually stopped doing that because there weren't very many, because these things are smaller, than ciliates, and so I think there's just no space in the gut or whatever for protists. And so what we learned from the bacteria, is that they are distinct from the environment. So if you look at the environment around them, the bacterial profiles and the meiofauna are different. But the different animals from the same environment tend to have indistinguishable microbiomes. So it's like they're not very different from each other, but they're different from the free-living things. And so our conclusion from this is that there are guilds, like functional guilds of bacteria that just are really like to live inside animals. And that the animals probably, like the idea that microbiomes are all about your development or whatever, probably came later. But it was driven by the bacteria who just adapted to living in animals and wanted to go find animals to live in. And then later, the animals adapted to that, and the functional things that people mostly pay attention to evolved from that. So what we're going to do now is move to bigger animals. So meiofauna are fun because not many people study them, but the bigger animals will have bigger microbiomes. And we've already found this, is that the diversity of bacteria and protists within slightly larger animals just goes up really rapidly. So I don't know. It's going to decide. I think the size is actually really, it sounds stupid, but I think the size is going to be the biggest factor. The more space you have, the bigger your community. There's someone at the back.
One more question at the back.
While he's walking, what's really interesting, Ramon, is when you do this in different places, you get completely different. Like this is mostly little, like crustaceans and things, but if you do it in a coral reef, you mostly get worms. And so we're gonna get like really interesting, like this. I don't work on animals, so this is fun for me, but I'm probably saying silly things. But it's gonna be neat to look at the distribution of the different kinds of animals in the protists. Well, we just do the light at night because it's a good way to guarantee you get lots of stuff. Yeah, but it's going to be, that is a bias too. You're going to attract certain things and not other things. So we're going to do it both ways.
Patrick, one question. Very nice talk. I was wondering about what you said, that these 11 losses in chlorophyll, that implies your phylogeny. What do you think? Do you think that perhaps there are other apicomplexans that can have chlorophyll? And how well sampled, according to you, are apicomplexans? Or now that you are going to do this new sampling, are going perhaps to appear sister lineages there? Thank you.
Yeah. So will other apicomplexans have chlorophyll? So the ichthyocolids is the obvious one, but they're related to corallicolids. So right now, I guess they don't, because from Javi's lab, the plastid genome didn't have any chlorophyll biosynthesis. Then the protococcidians are interesting because their plastid genome doesn't have any chlorophyll biosynthesis genes. But we did find the last enzyme in the step in its nucleus. So that's fascinating, because if the genes have moved into the nucleus, then they're going to be really hard to find. So we went back and looked at all the apicomplexan data from every group that's available to look for the nuclear encoded genes, and we didn't find any other than in the protococcidians. But if they're in the nucleus, they're way harder to find than if they're in the plastids. We also went back, there's a really big and diverse group of apicomplexans called gregarines. That's like the deepest branching group. And most gregarines have lost their plastid genome. So they still mostly have the plastid, but they've lost the genome. And so you can't tell with them what they've got. There was no representative. And similarly, the other deep branching group is Cryptosporidium, which has also lost its plastid. So there's no data from the very first branches. So we recently went and sequenced the plastid genome from a bunch of the only lineage of gregarines that's kept it, which are the Selenidium, or the archigregarines. And at first we thought we found the chlorophyll biosynthesis genes, but these are the things with like 9% AT. They're super crazy. And in retrospect, we didn't. So we now know they do not have them either. So currently, there's really no group of apicomplexan that hasn't got its plastid genome sampled that we know of, except for this mysterious apicomplexan X thing that nobody knows what it is. It's just a ribosomal RNA that we'd love to find. So I suppose if I had to guess right now, I would say no, but it would just be a guess. And as soon as someone finds a new apicomplex in the lineage, I'd say, one of the things I'd love to know is, does it have chlorophyll? But we got to find more new lineages first.
Cool. Thank you.
Hi, thanks for the talk.
Thanks.
About mouths, is there any information about groups 3 and 5, or are they still completely unknown?
Oh, I'm bad with numbers. We're getting lots of malv data in these plankton things too, by the way, but it's not, it's not tied to hosts, unfortunately. So, which one's 3 and which one's 5?
Yeah, you don't have them in most of your results.
I was looking for this one. No. Oh, right. Yeah. 5 is, no. Actually, the answer is no. We don't have any data from them. But fingers crossed. We're going up to Alaska again, and Liz is going to spend the entire time just doing single-cell transcriptomics from things that she sees are infected. And she's gotten really good at recognizing a protist or an animal that's infected with one of these things. So fingers crossed, but we don't have that data yet.
Thanks.
Okay, any other question?
All right. Well, thank you very much.
Thank you, Patrick.